Today, I'm going to explain The Gene, An Intimate History. This book, approximately 200,000 words long, will be summarized in about 24 minutes to convey its essence. How will genetic research change our world? Before we delve in, please click on the subscribe button to stay updated on more book essence from Daily Book Dive. A few years ago, American actress Angelina Jolie underwent a preventive double mastectomy to reduce her risk of breast cancer. This was a significant decision for a female celebrity. Jolie explained that her mother battled cancer for nearly 10 years before dying at the age of 56. She didn't want her children to experience the pain of losing their mother. Jolie's decision was influenced by rapid advancements in genetic science. Scientists have successfully identified the breast cancer gene, and women carrying this gene have an 80% lifetime risk of developing breast cancer. Jolie discovered through genetic screening that she carried the gene, and by removing her breasts, she reduced her breast cancer risk to 5%. Jolie's case demonstrates that for diseases like breast cancer, caused by genetic mutations, advancements in genetic science can help people better prevent these illnesses. Following this logic, if everyone could easily sequence their genes and scientists could identify all the genetic mutations that might cause major diseases, then we could each understand our potential genetic diseases and live healthier, longer lives. Is it that simple? Yes and no. Most diseases don't have a single, clear-cut causative gene like breast cancer. Many diseases result from a combination of multiple genetic mutations and environmental influences. Sequencing and screening genes won't immediately unlock the secrets of human life. Genetic research has only begun to uncover the complexities of the genetic world. The author of this book is Siddhartha Mukherjee, known for his Pulitzer Prize-winning work, The Emperor of All Maladies, a biography of cancer, which delves into the history of cancer's origins, treatment, and prevention. After writing The Emperor of All Maladies, Mukherjee was compelled to ask, if cancer is a biological anomaly, what about the normal variations and inheritance of life? Thus, The Gene, An Intimate History, was born, equally detailed, fascinating, and humanistic, and can be seen as a prequel to The Emperor of All Maladies. At the end of 2016, Bill Gates included The Gene among his top 5 favorite books of the year. Genetic research is a prominent field today, and life sciences based on genetic research are among the most crucial areas for future technological development. People hope that genetic research will discover cures for terminal illnesses and the secrets to health and longevity. Some also hope that genetic editing can eliminate disease-causing genes, add beneficial genes, and create new humans. Beyond scientific advancements, genetic research also has a strong humanistic concern. How much of each individual's makeup comes from genetics, effort, or chance? The Gene balances both scientific and humanistic perspectives, providing a new interpretation of fate and choice from a genetic viewpoint. I will explore 3 questions to summarize the key content of this book. First, how should we understand genes? Second, will genetic research challenge the theory of evolution? Third, what are the future focuses of genetic research? Part 1. What are genes? A professional explanation is that genes are the basic hereditary units controlling biological traits. Simply put, the life cycle of an organism, birth, growth, aging, illness, and death, is related to the information stored in its genes. Since genes store biological information and affect the organism's biological data, people might think that human genes contain the blueprint for constructing human bodies, determining all our physical, physiological, and psychological traits. In other words, mastering genes would enable cloning identical organisms. If you could deconstruct all my genes, you could replicate an identical version of me. The gene tells us that such understanding is too simplistic and binary. The world of genes is much more complex and chaotic. In reality, most genetic information resembles a recipe more than a blueprint. The key difference between a blueprint and a recipe is that a blueprint can produce precise, identical copies, while a recipe only outlines the steps, resulting in variations in the final product. Understanding genes as recipes helps us grasp how they influence our physical and mental development. Just like the sequence of steps in a recipe matters, with initial steps often having a greater impact on the final dish, genes also have hierarchical effects. For example, when baking bread, the first step is mixing the dough, specifying how much flour and water to add. The right ratio of flour to water yields suitable dough for bread. Too much water and too little flour result in a slurry. Similarly, early-stage genes often have a decisive impact on the final product, whether it becomes bread or slurry. An example is the gene affecting sexual orientation, which operates like a recipe during embryonic development, shaping sexual traits. The first step in this recipe involves a gene called SRY, which is crucial in determining whether an individual's physiological traits will be male or female, thus being called the master regulator gene. Activating or deactivating the SRY gene dictates the physiological traits as male or female. Biologists conducted an experiment where they implanted and activated an extra SRY gene in a female mouse embryo, leading to the development of entirely male traits, including a penis and testes. And the resulting male mice only showed interest in female mice. However, SRY only contains the first step's instructions in the recipe, subsequently affecting dozens of other genes that integrate internal and environmental influences, like hormones, social, and cultural impacts, ultimately shaping individual sexual orientation. While the SRY gene's impact in determining physiological sex is decisive, sexual orientation isn't solely dictated by the SRY gene. Variations in other genes in later steps of the recipe cause differences in degree and inclination. Earlier, we mentioned Jolie's breast cancer gene, which is also a gene located in the first step of the recipe. Its mutation essentially determines whether a woman will develop breast cancer, but when and what type of breast cancer the carrier develops is influenced by other genes and external environments. Not all diseases, however, have a clear master regulator gene like breast cancer. Schizophrenia, for instance, can be traced to dozens of related gene mutations, and if some mutations are not activated, they lead to varying degrees of mental illness. Genes not only determine physiological traits, but also influence genetic diseases. Beyond that, do genes affect personality, character, and talents? In other words, are all our physical and mental traits determined by underlying genes? Historically, genetic research has seen two extreme viewpoints on this issue. One side, environmental determinism, believes that a newborn is like random access memory, with a blank brain ready to download any operating system, with future psychological and personality development mainly shaped by culture. The other side, genetic determinism, argues that genes determine one's character, desires, temper, and behavior. Biologists and anthropologists have conducted numerous experiments to explore this. Results show that genes significantly impact human character and temperament, much more than generally imagined. One famous study examined identical twins raised in different environments. Identical twins originate from the same fertilized egg splitting into two embryos. Some twins are separated shortly after birth and grow up in completely different environments. Studying their differences years later provides insights into the genetic influence on individuals. Researchers found that separated twins developed many astonishing similarities, such as similar personalities, hobbies, and susceptibility to similar illnesses. Some studies even discovered that separated twins shared similar musical preferences, financial habits, and altruistic behaviors. Such research indicates that genes indeed determine not only appearance, but also personality. But does this imply genetic determinism? Not entirely. Consider why genetically identical twins, even when growing up in the same environment, become different individuals with unique personalities rather than exact replicas. A simple answer is environmental influence. Even when growing together, twins experience subtle environmental differences and numerous personal encounters. For example, one twin might develop diabetes, but the other doesn't have a 100% chance of developing it, although the risk is significantly higher than the general population. This is because multiple gene mutations and external environmental factors influence disease development. We can imagine twins as the same set of genes evolving into two different beings through a time-space machine. Genes determine part of their body and character, but individual evolution is intricately linked to external environment and unique personal events, like childhood illnesses or youthful accidents. The author metaphorically describes gene influence as genes writing a script for humans, leaving room for improvisation. This sentence profoundly portrays the meaning of life. Part of life is hereditary, but there's also space for chance and adaptation to external changes. Without variation, there would be no evolution, nor would there be us. To summarize the first viewpoint, genetic information is not a precise blueprint for organism development. It's more like a recipe, outlining the production steps without ensuring identical results. Genes determine our physical traits and influence our character and temperament. However, we must also consider gene interactions and responses to environmental changes. The author refers to the interplay between genes and environment as fate, and the individual's response as choice. While fate is mainly determined by heredity, it is also influenced by the environment, and different choices drive the world's diversity. Part 2. Will genetic research challenge the theory of evolution? Our traditional understanding of evolution comes from Darwin. You are likely familiar with Darwin's theory's two key points: natural selection and survival of the fittest. For example, giraffes evolved from certain herbivores, developing long necks because those with long neck genes survived a drought, passing on the mutation, which over thousands or even millions of years evolved into giraffes. But could giraffe evolution have happened another way? For instance, could a gazelle that stretches its neck to eat high leaves gradually pass this behavior to its offspring, leading to longer necks over generations? Can external environments leave such a memory in genes, and can this memory be inherited? The Gene, an Intimate History, recounts a real human story. In the winter of 1944 in the Netherlands, the German occupation restricted food and coal supplies, leaving millions in Amsterdam and surrounding areas facing extreme starvation and cold. Each person received only 3 potatoes a day, and thousands died of hunger, while survivors were emaciated. This human-made famine created a natural experiment, with millions facing sudden nutritional deprivation and physical and mental stress. Researchers found that children born during the famine were not only stunted in growth, but also prone to chronic diseases like heart disease, osteoporosis, and diabetes. In the 1980s, researchers revisited those born during the famine and found that babies conceived during the famine were more likely to develop chronic illnesses similar to their mothers, like obesity and diabetes. This is understandable, as fetuses in malnourished mothers would alter their metabolism to store more fat, potentially leading to adult obesity and metabolic disorders. Most strikingly, in the 1990s, researchers tracked the offspring of these individuals, the third generation, and found they also had a higher incidence of similar chronic diseases. This case shows that the genetic changes caused by the external environment can be inherited by the next and even subsequent generations. The genetic changes brought about by the Dutch famine indicate that evolution might not only occur through long-term gene mutations and natural selection, but also through the impact of significant environmental changes, leaving a memory in genes that is inherited over at least 2 generations. This might be another shortcut for organisms to adapt and evolve. Although this type of evolution results in chronic diseases due to overreaction to famine. Subsequent stem cell research also supports the existence of genetic memory. Stem cells, which can self-repair and differentiate into various cells, are common in embryos. A fertilized egg divides into an embryo whose cells can develop into different organs. Although these cells have the same DNA, their traits differ. Embryonic stem cells can develop into various cells. But skin cells, for example, won't suddenly become nerve cells or revert to stem cells. Is this development irreversible? One researcher tried converting skin cells into highly plastic stem cells and found that young mouse skin cells were more easily transformed than old mouse skin cells. They asked, why can't old mouse cells rejuvenate? They believed the answer lies in genetic memory. Responses to environmental changes, such as metabolic changes due to famine, are recorded in genes and cannot be erased. These genes with historical marks are harder to rejuvenate. To understand genetic memory, imagine each cell from an embryo having the same genes, like reading from the same book. However, different cells, due to environmental changes, delete some sentences, add words, underline some sentences, and bold others. Thus, each cell creates a unique interpretation based on the same book. Summarizing the second viewpoint, the core of evolution theory is gene mutation and natural selection through competition. As we delve deeper into genetic understanding, we find that genes' responses to significant environmental changes are firmly imprinted in genes, forming genetic memory. If this memory is heritable, it might become a shortcut for evolution. Part 3. Future focus of genetic research. Now, let's look at the future focus of genetic research. Genetic research is a prominent field in current technological development, with 2 main focuses: genetic diagnosis and genetic editing. Genetic diagnosis involves sequencing and screening an individual's genes to determine the likelihood of various diseases and predict physiological traits, character, preferences, and talents. Genetic editing involves repairing or replacing defective or disease-causing genes to prevent future illnesses or alter an individual's character, preferences, or talents. Genetic diagnosis and editing have significant potential in preventing and treating diseases. Over 10,000 diseases caused by single gene mutations have been identified. With the human genome sequenced, the next step is reducing the cost of individual genetic sequencing and screening. In the near future, everyone might know their disease-causing genes and the likelihood of various hereditary diseases. Genetic diagnosis is already widely used in prenatal screening, such as for Down syndrome. Future screenings will identify more disease-causing genes. Post-screening, there are two choices: abortion for fetuses with problematic genes or selecting only healthy fertilized eggs for implantation in artificial insemination. However, both options face ethical dilemmas, especially as more genes are screened and the impact of some becomes less significant. For instance, would you choose abortion if a fetus had a 5% chance of developing a certain cancer? Adults have fewer choices. Knowing your genetic screening results and identifying dangerous mutations still leaves limited options. The author coined the term pre-survivor to describe people seeking survival before getting sick. For example, Jolie's preventive mastectomy is a pre-survivor choice. Others might choose regular screenings and early surgery if cancer is detected. These measures can't guarantee disease-free life, but can reduce the fear of developing cancer. Theoretically, curing diseases by modifying defective genes in the body is possible. Biologists once used viruses as carriers to introduce modified genes into major organs, hoping to correct mutations and achieve treatment. However, such experiments in the late 20th century led to a tragic failure, with a participating teenager's death, halting the experiments. Despite setbacks, genetic research has advanced significantly in the past 5 years. Genetic editing has become a prominent area in biochemistry. A new technique allows biologists to precisely cut DNA fragments in genes, a leap compared to previous methods. Some biologists even predict that genetic editing, along with stem cell technology and nuclear transfer, could comprehensively repair human genomes. This implies that genetically modified humans might become possible soon. Hearing genetically modified often triggers instinctive concerns. And genetically modified humans raise several ethical, moral, and societal issues. First, our understanding of genes' impact on organisms, gene interactions, and gene-environment interactions is still in its infancy. Genes are complex, and their interconnections are not fully understood. Changes in some genes can cause a cascade of other genetic changes. The butterfly effect suggests that editing specific defective genes might have unforeseen impacts on the body. Second, defining good and defective genes is challenging. Not only do gene interactions influence outcomes, but the same gene mutation can have different effects in different environments. For example, many genes causing schizophrenia also potentially enhance creativity. Historical figures with mental health issues, like Van Gogh, Virginia Woolf, and mathematician Nash, depicted in A Beautiful Mind, might be considered mad in a common world but geniuses in the creative world. Similarly, some autistic children may excel in drawing or music, displaying exceptional intelligence. This might be nature's result of chance and variation. Third, creating perfect babies through genetic editing raises serious ethical and societal issues. Genetic editing can fix defects and add new genes, potentially influencing both physical traits and character. Suppose we can isolate longevity and intelligence genes. Should we allow adding them to embryos? if genetic technology is expensive and only affordable to the super rich? Would this exacerbate social inequality? Overall, genetic research helps us better understand ourselves and predict the future. Some scientists propose sequencing 100,000 babies and tracking them for life to understand genes' role in determining physiology, diseases, character, preferences, and talents. Recent advances in big data, cloud computing, and artificial intelligence provide biologists with better tools to create genetic avatars or virtual twins for larger samples, aiding in deducing possible outcomes from genetic recipes. Such research can predict individual risks for heart disease or gallstones and potential sexual orientation or alcoholism. Future genetic reports might list various potential risks and possibilities, like a guide for pre-survivors. In conclusion, genetic diagnosis and editing are key areas in future genetic research. Genetic diagnosis will enhance prenatal disease screening, while genetic editing can fix defects and add beneficial genes in artificial insemination. However, like genetically modified crops, genetically modified humans will face significant scrutiny. One thing is certain, with big data, cloud computing, and AI, our understanding of genetic recipes will improve, allowing better predictions of our future possibilities. To summarize, the key points of The Gene, An Intimate History, have been summarized. Let's briefly recap. The biological world is complex and chaotic, unlike the physical or digital worlds. Genes, as the smallest units of hereditary information, are no exception. We can't understand genetic information as a physical world blueprint. It's more like a recipe with step-by-step instructions, outlining production processes without guaranteeing identical outcomes. Gene-environment interactions determine our physical traits and influence our character and temperament. The recipe leaves room for adaptation to external changes, allowing diverse development for each individual. Deepening genetic understanding challenges core evolutionary theory. Research shows genes' responses to significant environmental changes form genetic memory, which can be inherited, offering a shortcut for evolution. Future breakthroughs in genetic research will focus on genetic diagnosis and editing, crucial for cancer and disease prevention. Genetic screening for embryos and periodic genetic reports for individuals will predict health and disease risks. However, if human genes can be edited like genetically modified plants, Mukherjee warns that life is complex and diverse, not easily regulated by precise editing. That's the essence of this book. The original book link is attached at the end for further reading. You can also click the share button below the video to share this book with your friends. Congratulations, you've just finished another book.